accomplish a couple things. So I have a few last things to say about plant growth. Unfortunately, there's a whole lot more to say, but I have to condense it into just a handful of things to say. And then leave some time too for any questions you might have on the final exam, which is Tuesday at 2, correct? Tuesday at 2 in this room. And any questions you may have on that. And then I also want to remind you that, over the weekend, even if you have questions as you're studying, I will be checking my email over the weekend if you have a quick question that I can help you with. So, but before we get into all of that, let's think about plant growth and development a little bit more. So we've covered the idea that plants can actually move through turgor movements, growth movements. We've looked at a couple of examples, that Nyctinasty idea where they will hold their leaves up or down at night. There's the phototropism idea where they grow towards directional light coming in through your apartment window. There's that seismonasty where the Venus flytrap will trigger its leaves to shut over the poor hapless fly. And so there's those types of ideas. And then solar tracking. Solar tracking is another one where they can use turgor movements to orient their leaves to the sun, depending on where it is in the sky. And then let me just cover this last idea that we have time for. Thigmomorphogenesis. This is more of like an overall growth shift. And so this is something that you can do with your houseplants if you want. It is the idea of mechanically disturbing your plants in a nice way. If you want them to be shorter and stockier, you can actually influence that by how much mechanical stimulation you give them. And so in this experiment, the one on the left was touched twice a day, that's all. And the one on the right was left alone. And so you can influence their shape, their morphology in this regard too. I did this really without thinking about it. I had 2 identical plants I bought and I brought home. I left one on the porch outside and one in the bathroom. And they were the same size at the start. And then I noticed the one in the bathroom didn't get any airflow at all, really. And it got very tall. The one out in the porch that was getting exposed to summer wind all summer long was way shorter and stockier. So you can just do it with wind too. Doesn't have to be a physical touch that you have to do with your plant. But so that term thigmomorphogenesis, thigmo is touch, morpho is to shift, genesis, the shape of the plant in this case, genesis is to create. So there are some other ideas that with plant growth that I don't have time to get into, but those are some of the highlights, just giving you some ideas of how the plants are responding to their environment. And sometimes it can be a very quick thing like the Venus flytrap closing, or it can be something a little bit more slower and gradual that takes some time, especially with growth movements. Okay, this is a topic I need to also discuss with you today, this idea that when you think about like phototropism, for example, it is not just any old quality of light that's going to cause some of these responses. It can be, in this case for phototropism, it's actually blue light that the plant is specifically responding to. And it is because it has a special protein or photoreceptor that we call phototropin. Okay, so phototropin is the photoreceptor that is designed to absorb blue light. And when it does that, for the phototropism idea, what's happening is as your plant is sitting in your windowsill, the phototropin protein inside its tissues is absorbing blue light coming through the window. And then it is telling, essentially, the auxin hormone to shift over to the dark side of the stem, like we talked about last time. And then as the auxin is moving down the dark side of the stem, it's stimulating those cells to grow. And then you get that bending pattern toward the sun coming through the window. And so this picture is trying to show that the phototropin, wherever you see this peak in absorption, where you see it going up like that, this peak. So you look down and you say, oh, that's around 450 nanometers blue light quality. And so this phototropin, that's how it evolved. It maximally absorbs the blue quality of light to then start triggering this response by the plant, in this case, to grow toward the light coming through your window. Simple experiments with little tiny seedlings, they can show that when the seedlings are really easy to show the bending pattern. So that's why they often work with coleoptils or just baby like corn seedlings. And you can see, again, different qualities of light, not much response until they did blue light, and then they could show. So then they started hunting for like, oh, well, what other kind of pigments are out there in the plant that are absorbing different qualities of light? And one of the other big things that phototropin can do for the plant, which is huge, is it will tell the stomata to open. And so if you think about a plant outside, when the sun rises and comes above the horizon and strikes the plant, it's a phototropin protein that will absorb the quality of light. It can also tell the stomata to go ahead and open for the day if everything's going okay, so they can keep taking in all that lovely CO2. So they started hunting in the plants for other pigments. What are photoreceptors, absorb light? They're usually a protein of some sort. And so they're like, it's a pigment protein complex usually. And so they're looking at, what else is the plant doing in terms of how it responds to sunlight? And so they came up with this term, photoperiodism, which is one aspect that they study quite a bit, which is basically asking the question, how do the plants figure out how long they're in sun versus how long they're in the dark? And so this period, the periodic flow of time and sunrise, sunset, light, dark, how are they doing that? And so that took a long time for them to figure out. And so what they started to appreciate is that the plants are keeping track of how long am I in the light, how long am I in the dark, and that will influence when they decide to flower. And I'm sure you've lived your life long enough where you've noticed some plants like to flower right in the spring. Other plants, they wait until the fall, and then they decide to flower. And so the question is, why don't all plants just flower in the spring, for example? And so they were really busy hunting this down. It took a long time. Here you're looking at poinsettias, which if you grow poinsettias for a living, you gotta know when and how to get it to flower so that they're ready to go and you can make a tidy profit around this time of year. So this is can be big business to understand this idea. So here, by the way, I can't remember if I told you this, but the flowers are really these tiny little things on the poinsettias. Those are the flowers. All the red, those are just modified leaves. That's not really part of the flower per se. A lot of people don't appreciate that, but okay. So what they started to realize is that a lot of plants can get grouped into these different categories. Here's one, short-day plants. They're called that because they're not going to flower unless the day length is shorter than some critical time period. And I'll show you some examples. So poinsettia is this way. They are not going to flower. If you look down on the bottom right here, it says they won't flower unless they receive 8.5 hours or less. And then on this image, the plant on the right got more than 10 hours per day, and you can see it decided not to flower. But the one on the left that got 8.5 hours or less, it decided to flower. And so they started to do all these experiments with these plants and saying, can we get them to flower? What allows them to flower? And so poinsettias fall into the short day category. And so that means if you want plants, the poinsettias, to be in the flower state when this time of the year rolls around, you have to time it just right and make sure that your greenhouses aren't getting any exposure to light after 8.5 hours. So that can get tricky, okay? And so knowing how to do this correctly can be big business. I have in the notes section, other short day plants, cannabis, asters, sorghums, ragweed, unfortunately, strawberries. Okay, so with this idea, they started to say, okay, yep, there are some plants that are short day plants. Then there are some plants that are long day plants, which is just the opposite. They're not going to flower unless the periods of light are longer than some critical time period. And potatoes fall into that category. That's why they don't flower till late in August usually. They don't flower right away in the spring. And so again, they started finding that there's a bunch of plants that fit into this category as well. In the notes, I have potatoes, spinach, wheat, lettuce, beets, those types of things. So they also discovered that there are plants that they call intermediate and day neutral. Intermediate just means it has to hit the sweet spot, not too short, not too long. Day neutral kind of is like, we're not quite sure how this, they don't seem to really care so much about tracking light versus dark. So it's not everything, every plant doesn't fit perfectly into one of these categories. There's a little bit of mystery still left, especially with the day neutrals. So you just have to give them enough light to just stay alive and do their thing. So day neutral, those tend to be the plants that live more right around the equator, where they're native to that region of the world. So tropical plants tend to be day neutral, which I guess kind of makes sense if they're sitting right at the equator. So it basically, all of this tracking, when the plant's like, how long am I in the light? How long am I in the dark? It's a way for them to figure out how to complete their life cycle and prepare. So especially plants around here, they have to prepare for winter. So they better get this right. Otherwise, they're going to be caught short when it gets cold and they're not ready for the cold. And so it helps them complete their life cycle in terms of making flowers, and then hopefully they'll get pollinated and set, seed and fruit. And so the hunt was on for what is it? What is this inside the plant that is tracking light versus dark? And it turns out it's a group of pigments called phytochromes. It's kind of an odd group of pigments. And it's a photoreceptor because it's absorbing light in order to do its thing. And so they find that the phytochrome comes in two stable forms. One that they labeled piece of R. R stands for red, because when it's in that form, when it's in the piece of R form, it's designed and ready to absorb red light. Then there's the other form. There's the other form called piece of FR. FR stands for far red light. That means when it's in that form, it is ready and able to absorb far red light. Okay, and then what happens is when one of these absorbs their quality of light, it makes them convert to the other form, which is really weird. And the way that they're converting to the other form is they're changing their shape, their physical shape. So that's weird too. So let's look at this diagram. So here, well, let's look at both. So basically what they're discovering is the plant is actually keeping track of how much piece of R do I have in my body? How much piece of FR do I have in my body? And it's really the ratio of these two, like how much of each. They're keeping track of that ratio. And that ratio is telling them I'm so many hours in the dark, so many hours in the light. And then that's going to tell them, should I flower? Should I flower now or should I wait? Should I go ahead and get ready for winter or should I wait? So up top is this idea of converting back and forth. So the piece of R form on the left here, that corresponds to what they've drawn down here in this image on the bottom, the way it's shaped there. Okay. So what's happening is when that piece of R form absorbs red light, it's designed to absorb red light, it actually causes it to shift its shape. So now it looks more like a capital Y in its shape. And then we call it the piece of FR form if it's in that shape. All right. So when the PR form absorbs red light, it physically shifts into the piece of FR form. Now that form, that capital Y form, it's designed, that form is really good at absorbing far red light. So if the plant gets hit with a bunch of far red light, it's going to take any of its piece of FR that it has and shift it back to the piece of R form. And you can do experiments all day long with this, where you hit the plant with red light, hit the plant with far red light. And you keep doing this back and forth, and you can see it just shifting. The pigments will shift back and forth. It'll physically change its shape. All right. And so what the plant is really doing is it's saying, how much piece of R do I have? How much piece of FR do I have? And based on that ratio, what do I have more of? It will tell them specific things about their light exposure versus their dark exposure. Now you see here, they're showing that when it's in the piece of FR form, then they show an arrow, and they're trying to show that this piece of FR form can interact with other proteins in the nucleus and ultimately start influencing gene expression. So that's why they say it's the biologically active form. It's because that's the one that tends to be communicating, so to speak, with the chromosomes. Okay, but really it's the ratio of the two pigments that the plant is really also tracking. Okay. Okay, that's kind of weird. Questions on that? Well, let's think about a couple of examples. Okay, let's go to this. So let's say we have a plant that's in the long day plant category. That means it needs some day length longer than a critical period in order for it to flower. So this donut shape of time is showing you the critical time period is the dashed line. And so if it's a long day plant, you want the light period to go past that critical period, which it has. So under this condition where the light exposure is longer than the critical period, that long day plant is going to flower. However, if you don't provide that long day, it's here in the second image, you can see that the light ends before the critical time period. So you didn't give it enough time, light time, and so that long day plant will not flower. Okay? So that's what we're talking about here with a long day plant. It's counting time. It's able to figure out, have I reached the critical period of light exposure or not? Have I gone that length of time? And if not, it's not going to flower. And that can be super critical for the plant. It has to be able to figure out, when should I flower if it wants to. I mean, when you think about why does a plant flower, it's so it can get seeds going and get its chromosomes into the next generation. It can persist and not go extinct. All right. So this is a huge deal. So you can actually play around in the lab with this, and depending on what quality of light you hit the plant with, you can kind of fake it out and have it flower or not flower. So if you keep this experiment going, so let's look at this third donut here. So here, if you just look at that, you'd say, okay, this is a long day plant. The light didn't make it past the critical time period. So based on that, it should not flower. But what the scientists did was they came in the middle of the night and they hit the plant with some red light. So remember now, red light's coming, sunlight has red light. So it's like the plant went, oh, I just got exposed to sun. And it starts to interpret it. It thinks about it like, oh, so I've been in the sun all this time. And so then it says, oh, I've met it. I've gone over the critical time period. I'll go ahead and flower. Okay, so you faked them out. This is in an experiment that you're doing this, okay? But then the scientist said, what's going to happen if we hit them with red light? Then we hit them with far red light. And they were really surprised to see that the plant decided not to flower. But then once they figured out phytochromes, it made sense, because let's look down here. Okay, if you hit the plant with far red light, so that's this lower swooping line here, so that means you're going to make the piece of FR form shift into the piece of R form. And so that means it's piece of R form. There's going to be more of that in the plant body. And that's going to tell this long day plant, no, don't flower. All right, so it's actually the phytochrome pigments that are getting messed with when you do these little silly experiments like this. So then when they came back and said, let's do red, far red, red. So if they see red light, that was their last light exposure was red light. What's happening is, oh, my red got, it should say red right there. It got cut off. Sorry. Let me put that. So this should say red. So if you hit the plant with red light, it's converting a bunch of its P sub R form over into the P sub FR form. So that's telling the plant again, oh, I have a lot of sunlight. I'm going to go ahead and flower. And so they were figuring out that it's this phytochrome and how much of the P sub FR versus P sub R does it have. And when a plant is experiencing true sunlight, I have it in one of these. Where did it go? When the plant is experiencing true sunlight, that last the sentence down on the bottom here, where it says, during most of the day, the red to far red ratio of light from sunlight is about 1.2 to 1. So what that's saying is there's a little bit more red quality of light that the plant gets hit with compared to far red light. Okay. So sunlight energy, there's a little bit more red quality coming at the plants compared to far red. And so they'll already have a little bit more red light coming at them from sunlight, which means they're going to have a little bit more P sub FR form naturally in their tissues, okay, compared to the P sub R form. So that's kind of their way of going, oh, I'm in sunlight. If they are getting this lots of P sub FR, that's telling them, oh, I'm in the sun. But if you get the opposite, where the plant starts to see lots of P sub R in its tissues, then it's like, ooh, I'm not in full sunlight. Because far red light, there's less of that in actual sunlight. And so it starts to tell the plant, I'm not in full sunlight. Maybe I'm getting shaded out. Maybe I'm in the dark. All right. And so the plant is always assessing these two, P sub FR, P sub R. And for the idea of flowering, it is super important that they get this right.
So on the previous slide, red light is that clear before? What's light on?
So when the P sub R form absorbs red light and you get this shift where now the phytochrome becomes P sub FR, that's called the active form. And so that usually is telling the plant, hey, you're in sunlight. And then depending on the plant, in this case, for a long day plant, it'd be, hey, you're getting good sunlight. Go ahead and flower if you're past this critical time period. Okay. So that's why where you see that wherever it sees red as the last quality of light, it goes ahead and flowers. So it's kind of weird.
So say I just plans on flowering schedule and then forget to turn the light off on that and stay on for extra hours. How is that?
Yeah, that can mess with it. And they've even found moonlight, like bright full moon, can mess with plants. So, it's not even just natural artificial light. It can be something like that. So, and then that makes you think about, well, what about like landscaping lights and things like that, and they say, yeah, if it's bright enough, most nighttime landscaping lights aren't bright enough, they've found, but some are. So, there's some of those bright, like motion activated lights, So even that, like if someone walks by and they have this burst of white light on plants in your yard, for example, it'll mess with them. So the greenhouse here with all the poinsettias, I can't remember if I told you that story. I heard it on NPR podcast thing where they were interviewing a poinsettia businessman. That's what he did. He grew poinsettias for Christmas time, and he had huge greenhouses like this. And he had a worker come in the middle of the night and accidentally turn on the light. And almost all the plants in there did not flower in time. And so that he lost a lot of money. And the interviewer said, oh, I bet you fired that person. And he said, actually, no, I didn't. He said, because that person will never forget that. You know, the person was so upset that happened. And he didn't like, you know, penalize the person or make them pay for it. He just said, I know he'll never make that mistake again. So I didn't fire him. I kept him on. I thought, wow, that's pretty cool. Good way to think about it. So, but yeah, it can make a big difference.
That probably depends.
Yes, absolutely. You got to know your plant. And like I said, it's not always cut and dried. Like some plants don't seem to fit very neatly into some of these short day, long day. So you have to do a little bit of research and see what you can do with that. But like, you know, a lot of the commercial plants, they've figured this out. They know it's a short day, long day. And so when you start working with some of these plants, they, you know, for business, they'll have all that down. So here, this is from the book, just showing that you can get different shapes of the plant too, as well as the flowering idea, depending on the quality of light. And so here, that plant was grown with pretty much high red light. So remember, back in the controlled environment egg slide, when I said that red and blue light keeps plants compact, right? I said that it inhibits stem elongation, plants are shorter. So let's go back to that image. So that's what's being shown on the right here, okay? So far red light and blue light give that shorter compact look. The one in the center was just under normal sunlight. And again, it's fairly compact, but you can see how if you look at the far right one to the center one, you've made it even more compact when you've taken out the far red light. And then the one on the left, you know, it's much more spindly, tall. That one was pretty much dosed with far red light. So when far red light, like when you go back to that slide from the other slide set, remember I said far red light makes the stems elongate and there's fewer branches. Okay, so again, how the quality of light, the type, and duration, all of that will impact the shape and design of the plant. Where did it go now? There. Okay, so understanding, and again, if you grow plants indoors, this is very easy to influence. You have a lot more control over the light than, if, of course, if you're growing plants out in the agricultural field. And so you can really design the shape of the plant as well as how does, you know, when to get it to flower, all of that. And so it's pretty cool once you understand the plant. This is from the book. This is just a different graphic trying to get you to think about what happens. If you, in this case, they put it onto one, they're showing a short day plant versus a long day plant. And the critical day length is this dotted line here. And so they're just trying to, this is basically trying to look at, well, they're only showing a short day versus a long day. So if that graphic helps you figure this out a little bit more. But basically what we're thinking about, you know, with sunlight energy coming, if you think about plants outside, that sunlight energy has a little bit more red quality of light. The plants will get dosed with that more than the far red. However, if you think about a forested ecosystem, so that top right image is trying to get you to think about, okay, if you have like the canopy story of plants, which are the tallest, and then you have your understory, and you can have several layers of understory plants. So what happens is this blue line over on the left graph is showing you when the upper canopy gets hit with light, they're getting hit with a lot of quality of light. But what happens is those upper canopy leaves, they'll start to hog or, you know, take certain quality of light for themselves, right? Because in order for photosynthesis to occur, they actually absorb red and blue light. So if a plant is absorbing red and blue light, that means they're keeping it in their tissues. It's not going to the understory. Plants don't care as much for the far red, so they're like, I don't care about that. I'm not going to really absorb much of that. I'll just let that pass through to the understory. So in natural systems, you know, forested ecosystems, if a plant is getting hit with a lot of far red, that's telling them I'm in the understory. I'm getting shaded out. There's something above me that's hogging the red and blue light, so to speak. All right. And so it can be a cue to them to figure out where they are in the canopy system of that forest. And then that is a cue to help them adjust to that and deal with it. Some understory plants are totally fine with that. They're like, I don't care that I'm getting shaded. I'm designed to capture far red light okay and survive and do what I need to do. But there are other plants that are like, yeah, I need better light. And then they might have to shift their gene expression and say, grow, I need to get taller, someone's shading me. All right, so it really does depend on the plant, but basically they're using these qualities of light that they're getting access to. That's important information to help them decide how should I behave, how should I respond to them. Okay, so there's a lot going on. You're out walking in the woods. Those plants are very busy trying to figure out where they are in the scheme of things here and then behave appropriately. So this picture on the left is showing the blue light. I mean, the blue line is like what the upper canopy would be getting access to. And then you can see the other lines below it are what kind of get filtered through. And so you can see, you know, they're showing the far red and the phytochrome far red absorbs the far red quality. The piece of R4 gets the red quality. So not only are plants absorbing red and blue for photosynthesis, but there's these other qualities that they can absorb, you know, like the phytochromes are absorbing their far red. And then you have the phototropin absorbing blue. There's some other pigments I've talked about that absorb their qualities of light. So there's several different categories of pigments in the plant, photoreceptors in the plant that will work with these different colors of light. So again, understanding this when you work with plants indoors, and you can really play around with this and have kind of direct in a more controlled way what's going on with that plant in terms of how they look, when they flower, all of that. So it's kind of fun to think about. Okay, some other things about plants. You may have heard these terms. I just wanted to clarify. I hear these terms all the time, and during gardening season, especially if you look on seed packs, they'll tell you if it's a determinate plant or an indeterminate plant. And that has a specific meaning in the realm of botany. So if it's a determinate plant, what's really going on there is that you have flowers that are going to form at the very tip of the plant. And so that means it won't grow any taller, essentially. It's shifted totally into flower formation. So that's what they mean by determinate. Indeterminate, on the other hand, the flowers aren't right at the tip of the shoot. Instead, they're in the axillary bud areas. So the plant theoretically can keep getting taller and taller and taller while it's still flowering. And so a lot of plants are indeterminate. And a lot of trees are indeterminate, for example. So here they often focus on this with tomatoes. You'll see that a lot on tomato packages, the seed packages, they'll tell you whether or not you have purchased a determinate or an indeterminate one. And it depends what you want. Indeterminate tends to be a little bit more, you get fruit over time rather than all at once. And so that's what they're trying to show here with these images that I grabbed, just trying to show the difference between. And so it can matter to you depending on how you want, in this case, to grow your tomatoes and when do you want your fruit to form. Want it all at once or staggered. But I see a lot of, you know, different readings and things where they talk about determinate and indeterminate, but they never really explain it. So I wanted to show that difference to you. Okay, and then a little bit about this idea that plants do go through puberty, so to speak. And so, and then you have the annuals, biennials, and perennial terms that I'd like you to be familiar with. So annuals means they got to do it all in one season. They got to get it all to happen all the way to seed, setting seeds. Biennials means they usually grow vegetatively for 1 year and don't flower, but the next year they will. So it's like they have to go through a little bit of a juvenile period the first year. Perennials means that they might die off, or part of them might die off, I should say, in the fall around here, but they'll come back in the spring. You don't have to replant, they're just there. They're usually underground stems or roots that pop up to root tissue. So perennials are awesome in your landscape because you plant them once and they'll always come back. And then remember that, plants have some similarities to humans and other animals in the sense that they go through juvenility, or they have a juvenile phase where they're growing, but they can't have any kind of sex that would be successful. They don't form flowers yet. There's nothing going on that let them go into that. They have to pass through that juvenile phase, and it can vary from plant to plant how long that's going to be. It can be weeks, months, decades. It just depends on the plant. So you have that juvenile phase, then the maturation means that they can start going through reproduction. Senescence means they're starting to go into the twilight of their years. They're starting to age out, and parts of their tissues are dying. And then ultimately we'll have the death phase. And so also with juvenile phase, I wanted to point out, I've gotten tricked by this over the years. I have to always remember this. So someone says, what kind of plant is this? I need to know how old it is. And then sometimes I have to look it up because plants go through different stretches of time to be juveniles or into maturity. But these leaves, look at the difference in the leaves design. It's the same plant. It's just whether or not it's a juvenile stage of its life or in a mature stage. And you can see super different leaf morphology sometimes. And so that has tripped me up more than once, where I just forget to think about this idea. Yep, ivy on the top, right? So, and sometimes it's super dramatic in terms of the shift, and sometimes it's a little more subtle. So just be aware that sometimes they can look very different compared to the adult. And it's always a good question to ask. Do you know how old this plant is? So, and then meristematic tissue can shift quite dramatically. I don't have time to go into how the flowering is induced in any detail, but that shift from just normal tissue growth into flowering growth is pretty dramatic. Lots of gene expression shifting going on, and they have to get it right. Obviously, they can't flower in the wrong time of their year, especially when you think about plants around here. So they're paying attention. Certainly, they have phytochrome pigment that they're using, but they also pay attention to temperature. What's the temperature I'm being surrounded by? And so all of that, hormones, of course, are hugely involved in making sure that the plant is assessing things correctly and responding correctly. And so it is complicated, and each plant has slightly different requirements in order to decide when to go ahead and flower. But they're not, I just wanted to make sure you understand, they're not just paying attention to phytochromes and what's going on with that. There's a lot of other things that come into play here for the plant. And it makes sense that there's lots of kind of signals that the plant is listening to so they don't get it wrong. Because if they depend only on one thing and they get it, it's easier to make a mistake, so to speak. And so they'll be looking at a variety of ways to determine, should I flower or not? Should I go into dormancy or not? And should I break dormancy? That plants can get tricked by that sometimes. Okay, so we go on and on with plant growth and development, but we got to stop. So that's the end of the information on plant growth and development and how they respond to their environment. And so and now I'd just like to open it up to any questions you may have on any of the exam content. So you got about 10 minutes here. What else should we go over? Is there a concept you want me to go over one more time before we have the exam on it next week? where did we end off on Wednesday? We went through the light reactions, the C3 cycle. Pardon me? Minerals and nutrition. Yeah.
I don't know, if you after, there's a final, like, is reviewed, and then it goes through process, and then basics, and then, like, it takes it to another, like, reduce. What?
Yeah. Yeah, good question. So they don't know. So I have looked and looked and looked. When I go to plant biology conferences, I ask experts that study this in detail, you know, and they can't tell me. So no reading that I have found will do it. So let me go to, so this PQ in the Q cycle here, it grabs one electron through this process, and then it's waiting. It needs one more electron. And so it waits for another PQH2 to come along, which is being shown here. So here's your second PQH2 coming along, giving off that second electron. And then it grabs two protons, and then they show it here releasing, but they don't know exactly. It's really hard to track, like how do you keep these separate from each other? And so they kind of get vague at this point. The closest I've seen someone say anything about it was they say, well, we think that it also plays a role in, like, it'll dump its protons, it'll go to another cytochrome B6F and do.
So it's just like the establishment.
Yes, yeah. And so, but basically the upshot of the Q cycle is that it is a mechanism to allow for more proton pumping through the light, you know. So if you you mentally take away the Q cycle, you don't get nearly as much proton pumping through this process, you know, just the rest of it. So they say the true benefit of the Q cycle is that you get this extra protons disappearing from the stroma, showing up in the lumen. And that's a good thing, because once you get that proton gradient established, I wish the artist had been a little bit more liberal with lots of protons over here. So protons getting build up in the lumen compared to less in the stroma, then that's your proton gradient. And that's great, because now those protons, by the law of diffusion, they want to diffuse and even out across that membrane, but they can't go through the regular membrane because they're charged. So they have to go through that ATP synthase. And the action of them flowing through that ATP synthase actually causes these subunits to move and rotate. And then the ADP gets phosphorylated. These two come into close contact with each other through that rotation. It's bizarre. Oh, so yeah, it's the term, let me pull, it's the linear photophosphorylation. That's essentially what I just described, is this idea of getting a proton gradient getting established, where you have, as the electrons are cooking along down the electron transport chain, you get that proton gradient established with PQH2, the Q cycle, water's donating protons into the lumen. So all of that sets up the stage where you get lots of protons in the lumen. And so this whole process they call linear photosphorylation. That's the term I want you to know. That's how I'll refer to it in the example.
And then like
So photosystem 2, so photosystem 2, photosystem 1 are the two photosystems that actually absorb light. And then you have the ATP synthase, another big one, and then the cytochrome B6F. So those four are, you can see they span the entire width of the membrane. So we call them transmembrane complexes. So they kind of stay put. They're so big, they just kind of sit there. And then you have other players in the game that are mobile, they can move around. And so that's the PQ, the PQH2, the PC. Those are, so, whoops, those are our mobile, we call them mobile carriers. So PC, and then these two, FD, F, and R, technically those are mobile carriers. So they move around. So they grab electrons from one player, move around and deliver it to the next player. And so, but the big four just kind of sit there. They're too big to. No, abbreviations. Like I tend to abbreviate everything. I don't write out, like, if I say something about plastocyanin on the exam, I'll probably leave it at PC rather than write it out. Same like with FD, FNR, PQ, and then like photosystem, I usually do PS2, PS1. So that's fine. You don't have to write them out. Um, let me go to this. Let's see. What else? Let's see. Oh, stop. Why is it doing that now? Okay. Other questions? So I think we left off. Uh, what I think we left off with the mineral. Okay, here's the mineral nutrition. The characteristics of the soil, that cation exchange idea. So once you have minerals getting into the water of the soil, but you have to figure out how, like know the basics of how that happens. That's the cation exchange process. The anions are just always in solution, but then you run the risk of them going down into the groundwater if you over fertilize. And then we talked a little about macro, micro nutrients, and then immobile versus mobile once they're inside the plant. Questions on any of this? Yes. Basic symptoms, like you could use, if you see a picture, or could you see if it was described in words, would you know what chlorosis meant? Necrosis, model, general, yeah, the basics of that.
Do you need to know what deficiencies are among?
Yes, please, that little chart there on slide 23, which ones are mobile versus immobile. And the way I have had students tell me they do this is they just memorize the immobile and call it good. Because then whatever else doesn't, they just memorize that list. If it's not on that list, it must be mobile. That's a good way to do it for this class. I'm fine with that. Other questions? We talked a little bit about biological nitrogen fixation versus the Haber-Bosch process. Biological nitrogen fixation is so much cheaper in so many ways, more efficient, but unfortunately only a handful of plants can do that. Well, it's not the plants, it's the bacteria that hang out with the plants, but unfortunately there's very few symbiotic associations that can do that. Well, they're working on it. I'm trying to get more plants to do that. What did we do after mineral? What was the next topic? Soil management. Whoops. No, where is it? This slide set's pretty short, but a few key ideas. What can go wrong? Erosion and compaction and things like that. Anaerobic, really not a good thing. Any questions on this slide set? Thinking about cover crops and organic amendments. Some key differences on how the plant will, if you do just regular inorganic fertilizers, the plants pretty much just take that up straight. And so it can be a very quick burst of nutrients for them, whereas organic amendments tend to be a little bit slower release, slow and steady, longer influence. Any questions on soil management? It's pretty straightforward. Just spend some time going through it. What's after soil management? Okay. So that one is looking at actually a few topics. Including quorum sensing. This idea that for biological control organisms, plant growth promoting organisms, how do we do that? Vermicompost, organic amendments in general. We looked a little bit at this concept of using microbes to help the plant. Gotta kind of think it through carefully because it's not quite so straightforward. The white slide set is looking at the idea that in this particular case, having monocultures actually improved biocontrol. It made the bacteria more aggressive against the pathogen, the good guys against the bad guys. So it's more complicated. And then a little bit about pest control. Hopefully you'll never have these on your plants. But if you do, we'll have a way to diagnose. Questions on this slide set? Okay, so like I said, over the weekend, Monday, I'll be checking email frequently. So if you have last minute questions, please remember to get sleep. You need sleep to kind of get this all straight in your head, right? So you need some quality sleep. Take care of yourself. And I'll see you on Tuesday at 2.